Hello everyone, welcome back to my channel. So in the last class we have discussed about the monohybrid growths. So today we are going to look upon the topic called the test growths. So why the test growths is done? Test growths is done to find the genotype of the test organ, to find the genotype of the parent. Okay, we are undergoing the test growths to find the genotype, the correct genotype of an organism. We may be knowing about the phenotype, whether a plant is tall or a dwarf can be seen from the physical appearance. So, how will we know about their genetic makeup? For that, the test cross is done. For that purpose, we are doing hybridization experiments with the recessive parent. The main point is the recessive parent. We are crossing the F2 generation with the recessive parent. Okay. That is, we know that tall plant will be having two genotypes, that is capital T and small t. Capital T and capital T small t. This is homozygous and this is the heterozygous. So we should know what is the genotype of the current plant. We have got a plant but we don't know what its phenotype is. For that we are using it with a test growths. For that we can see the case 1 option. In case 1 we will be crossing this capital T into small t. So what will happen? One gamete will be drawn and what will be the result? Capital T, small t. Okay. In the second case, let it be a heterozygous one, capital T into small t. When we are writing the gametes, we will be getting like this. Isn't it? So we will be getting capital T, capital T, small t and small t. So in both cases we can see that here there is 100% tall plants we can see. Isn't it? But what about in this case? Here we can see only 50% of the total plants are tall. Because this one is tall, okay, but this one is a dwarf one. So if we see as a generation, we are getting only 50% of the tall plants. If we are getting only the 50% tall plants, the parent will be a heterozygous one. And if we are getting 100% of tall plants, it will be a homozygous. This can be easily seen with this point itself. Capital T will be having 100% things. And what about the heterozygous one? Only 50% will be here given. So this is the main focus of the test growths, because by this process we will be able to find the genotype of the parent only with crossing with a recessive parent. If we cross a plant with a recessive parent or a dwarf plant, we will be getting the result. If we are getting 100% tall plants, then the parent will be a homozygous dominant, and if we are getting 50% only of the population, then it will be a heterozygous dominant. So that's all for today. Hope you have got the point on the test growths, and if you have any doubts regarding this session, please comment on the comment section. Thank you and see you in the next video.